here and getting educated. So I'm probably going to talk fast, and I'm a little nervous, so I might talk even faster. So today I'm going to talk about cannabinoid science and cannabinoid lore. I'm a senior innovation scientist at Canopy. We used to be EBU in Evergreen and had a multidisciplinary team of scientists doing this work. So, with that being said, the agenda. I'm first going to talk about just a general education, general overview.
Can't hear you.
You can't hear? How about now? Better? Okay, so first I'm going to talk about just a brief overview about the science and pharmacology of cannabinoids, what psychopharmacology is, and how it affects people, separation science, how we get to purified compounds, how important the formulation of these compounds once they're separated is in producing a medicinal product. And I'm not having good luck with this at all. And then kind of the cosmic debris, what people are saying about it, all of the centuries of lore, and what we're doing to turn that lore into real scientific data. Finally, quality versus quantity, what are you buying and consuming, and I'll give you some takeaways. So first of all, what is a cannabinoid? A cannabinoid comes in several classes. So the first one would be just regular cannabinoids. They're defined as a compound that binds to the CD1 or CB2 receptor. So there are several subclasses of cannabinoids. One would be phytocannabinoids, and one would be endocannabinoids. This is fun. There we go. So we have cannabinoids in the middle, and then phytocannabinoids are any cannabinoid produced by a plant. They are always made as acidic compounds, not the neutrals. And then we have endocannabinoids, which are produced by the human body.
Yeah, your computer keeps going to sleep. That's how it's doing it.
It's not my computer. So we
it's
have juice for it, a power
yeah,
pack,
I would.
or I could just use my computer.
Yeah, I have the computer for comment.
Give me 2 seconds.
Yeah.
Sorry about that, everybody.
This one. Thanks. So I could keep talking, actually. So endocannabinoids were discovered very recently. 1992 is when they were first discovered. So they haven't been around a very long time with regard to, scientific studies of pharmacological agents. There we go. It's like whack-a-mole, it keeps moving. And,
Is it stable on your computer?
Not really. I think it's the dongle. All right, I'm not even going to touch it. Don't do it. This cord. Okay,
No more touching.
no touch. All right. Oh, it's loving me. So endocannabinoids are made by the human body. A couple examples are anandamide and 2-arachidonyl glycerol. There could be many others. We just haven't discovered them yet. It's quite difficult to discover endocannabinoids and other endogenous receptors type activators. So what is a receptor? A receptor is basically a protein that is expressed by your genetic code here. So here we have our number 6 chromosome that expresses CB1. This is a computer rendering of CB1 and what it actually looks like, the protein. CB1 lives inside the membrane of a cell, and it has these little curly loops, and it's coupled to what's called a G protein, and they're called G-coupled protein receptors, GPCRs. The things like your taste buds are also GPCRs, and they tend to be promiscuous receptors, meaning they will bind to more than one compound to be activated. So inside the cell we have this G protein. Outside the cell we might have an agent that comes in and binds to it and activates it and causes a change inside the cell. The change is very kind of complicated. It's literally like a Rube Goldberg machine biochemically. So on the left here, if you could see it, there's quite this machinery of biochemistry that in essence causes calcium ion to flow into the cell. When that calcium ions flow into the cell, we can measure that flow in real time. So with those kinds of measurement tools, we can determine activities, effective concentrations, and things like that. So, what is a receptor? A receptor is basically a lock and key type of mechanism, and you can think of in your cells and in your body as a sea of keys floating around in cytoplasm outside your cells. And this key will fit to a very specific protein or a receptor. So they're highly specific, and there are many, many thousands of them. So when all these keys float around, you can imagine just the probability of the right one bumping in and activating it, and this is all happening at like nanosecond time scale. So it's very, very rapid and quite amazing. Any ideas? So how it actually works, it's lock and key. So we might have a key, an endogenous activator, say it's a hormone or a neurotransmitter, and it will fit into the lock and open it. When it opens it, it causes a couple responses. There's a molecule called CP55940, which was made by Pfizer in 1974, and basically this will activate CB1 at 100%. So we use it as a marker for 100% activation. It's a synthetic cannabinoid, and it produces a full response, if you could see the graph. From that, we might use an antagonist, which is, say, a phytocannabinoid, or a synthetic cannabinoid, or anything that's not made by the body. And here it's represented by this little paper clip, and it will also open the key, and it might cause activation, but not 100%. It might be 50% or 25%. So all these different compounds can activate the response of the receptor at different levels. So we use CP55940 as kind of the standard mark for 100%, and we compare all of our other actives against it. Then we might have something called an antagonist or an inverse agonist. And this is like jamming a stick into the keyhole. And when you do that, nothing else can combine, so it kind of occupies that binding site, and it's a neutral agonist, so nothing else can go and activate that because the site is occupied. It can also do something a little nasty and make an inverse activation where it goes negative. And we see this happen like with K2 and spice, the kids that smoke this and end up dying or gravely ill, and it's because there was some kind of inverse agonist acting on it. They can bind irreparably to the receptor and cause a lot of problems in your biochemistry. So those are kind of how the cannabinoids interact with the receptor. So how many cannabinoids are there? Well, it's like alphabet soup. There are 120 that have been characterized by Mechoulam back in 1967. So he did a lot of extensive research and found 120. The most common are kind of on the right. These are what I see in hemp and cannabis strains that I can get at regularly and easily. There's about 20. And they come both in acidic and neutral forms. Plants only make acidic forms. For them to become neutral, you add heat and it loses CO2. It's called decarboxylation. And this little CO2 group will pop off a CBDA and we'll have CBD. So in essence, neutrals are a degradant of the acidic forms which are made by the plants. CBN is a degradant of THC, and these are the ones I see most commonly with the strains that I've been extracting for about the past 10 years. So the ones on the right here, these are the most common. And they're like CBD, CBDA, THC, CBG, CBC, THCB, and other variants. The variants are real simple. It's just a matter of how many carbons are in the little tail of a molecule. Nice try.
You're doing great. Keep it up.
So THC or any of these cannabinoids, they have the a 5-carbon tail, whereas the THCV will only have a 3-carbon tail. This raises some interesting questions about other variants that might be like a C1 carbon, just a 1-carbon tail, or even number carbon of tails, like 2, 4, or 6. And those aren't seen anywhere in nature, but they could be synthesized and we could test their pharmacological activity and things like this. So, this is pretty hard without a slides. Measuring receptor activity, and I have this awesome picture. In fact, this might be better, yeah. In any case, is there any way we can get these slides working? This is really good.
Just keep talking.
Okay, keep talking.
Just keep talking. Just use your hands.
Use my hands. So, oh yay, look. So we measure these, the activities of these compounds using what's called CHO cells. And CHO cells are mammalian cells that we have genetically modified. I showed you that little genome, our little gene. Well, we take the gene for our human receptor and we genetically modify these cells so that they express our human receptor on the surface, and they express a lot of them. So we have them overexpress the cells. And what happens is when we apply a compound and it activates it, we can, calcium ion flows into the cell. When that happens, they actually glow, so they fluoresce, they turn green. And they'll start out dark, and then they come up, and then they intensify, and then they fade back out. And we measure that real time and can determine numbers like the EC50 and IC50. And these are pharmacological numbers that are important that determine like what is 50% or the concentration of a drug that gives 50% of the maximal response. That's called an EC50. Well, receptors can either be turned on or turned off. So if you're going the other way and want to turn it off, then we call that an IC50, the inhibitory concentration that reduces it by 50%. All of these studies are called pharmacodynamics, and it is the study of the biological effects kind of at the cellular level of drugs and therapeutics. For each receptor, we have to have a unique cell line. So currently we have about 8 cell lines. There's CB1, CB2, TRPV1, which is the capsaicin receptor that senses spice in your tongue and on your skin, and also serotonin receptors. There are many, many other receptors that cannabinoids have activity with, and those are being uncovered almost weekly with all the science being brought to bear on this subject now. So, it's very expensive to raise these cells. They have to be grown in an incubator, petri dishes, and loved and coddled and kept alive. We use what's called a flipper, which is a fluorometric imaging plate reader. And this reads the photons of light that are emitted and can quantify it. So it's very accurate and very powerful tool for pharmacologists to determine what they're going to make in the next drug. So, with pharmacokinetics, it's a kind of side study. It goes with pharmacodynamics, and this is really the study of how drugs get into your system, how they're distributed, metabolized, and excreted. So we have this famous acronym ADME, absorption, distribution, metabolism, and excretion. And it goes kind of hand in hand with the area under the curve, the AUC. So when you give someone a dose of medicine, say it's a pill oral, it goes in, how is it absorbed? Is it absorbed in the stomach? Is it absorbed in the intestine? What happens after it's absorbed? Is it processed by the liver? Are the metabolites of the drug actually the effective ingredient? Where does it go to do its magic? And then how does it get out of your body? The area under the curve part tells you how it moves, how quickly it gets in, what the maximum concentration is, Cmax, how long it takes to taper off. And here we have what's known as a therapeutic zone. So there's a specific concentration that gives the desired therapeutic effect. And we want to keep the concentration in that zone. And so when you multiple dose, you kind of get this jagged line and you keep the concentrations to where it has a positive impact on your ailment. So this brings me to bioavailability, which is a very, very important term that you don't hear much discussion about today. And it's kind of the critical question to ask before you ask, how much do I take? So saying how big a dose of CBD should I take is kind of putting the cart before the horse. We need to know the bioavailability of this formulation before we know how big a dose to take. Bioavailability is defined by how much actually gets into your blood of an administered dose. So IV administration is 100% bioavailability, direct access to the bloodstream. Whereas, for an example, CBD isolate, if you were to take 100 milligrams orally, only about 6 milligrams is going to get into your bloodstream, giving it a 6% bioavailability. So things that aren't soluble in water, they're hydrophobic, like waxes. They have very low bioavailability. They don't mesh well with biological systems. They're not soluble in water, but they have great activity once they're there. How do you make them water soluble? How do you get them into the system? CBD isolate is definitely very low bioavailability. So 100 milligram dose, you get 6 milligrams. If you apply technology to increase bioavailability through encapsulation or emulsions or solid dispersions, there's a whole number of technologies to use. And say we were to do a 50% bioavailability form, now I only have to take 12 milligrams of the formulation to get that same 6 milligrams into my system. So formulation is everything. It goes hand in hand with both the route of administration and how much of a therapeutic dose you need to get in. This helps you get it in. So we'll switch gears to psychopharmacology. What is that? Psychopharmacology is kind of the impacts on mood, sensation, behavior. So these can be measured using visual analog scales. And you might have seen one for pain. It's the smiley guy at 0 and the crying guy at 10. That's a visual analog scale for pain. We can develop all manner of scales to measure lots of different feelings, like energy or lethargy, happiness, sadness. And we would measure pre-dose and post-dose time points to get an idea of the psychopharmacological impacts of cannabinoids. So with that being said, what about the entourage effect? What about multiple compounds that come together and 1 + 1 isn't 2 anymore? 1 + 1 is 8. They have synergy. They make each other work better. And in cannabinoids, there's a very strong belief that the entourage effect is really what contributes to the enjoyment of cannabis use. What illustrates this is Dronabinol, which was pure THC, is a pharmaceutical that's been out for a long time, also Marinol. It's got a very, very low potential of abuse. Why? Because pure THC doesn't really imbue a desirable intoxication. So it has very low potential for abuse. However, if you combine it with terpenes and CBD and other cannabinoids, now all of a sudden it's a very desirable effect. And we see that because of the popularity of cannabis use worldwide. So why is the whole plant give a better effect than an isolate or a pure compound? And it's really because of the entourage effect, which is Okay, thanks. Which is due to drug interactions. Drug-drug interactions basically can change the way a drug works, the way it's metabolized, and the impact it has. It can make it more effective or less effective. And so this is why drug-drug interactions are kind of tied to the entourage effect theory and the poly-pharmacology theory. So in today's pharmaceuticals, it's really monopharmacology. You have a single agent acting on a single receptor to alleviate symptoms for a single ailment. So it's still very complex to get that all passed through the FDA. But if we look at poly-pharmacology now, we have multiple active ingredients working on multiple agents. It becomes much, much more complicated. And I just love this graphic. This graphic is a perfect illustration of what polypharmacology is like. So we have this therapeutic with lots of actives, and they fit into all the gearing of our biochemistry very intimately and precisely. And we want it to fit in so that it doesn't seize up the machine. We need all these gears to keep rolling. So all the little gears and ratios of compounds and what this represents, it's very complicated and very unpredictable as far as what it will do to the whole organism, and will it go in and jam a gear and make the machine not work. These might be seen in negative drug-drug interactions. So this graphic is very indicative of what we're trying to do. We're trying to find the combination of cogs that fit in and make a specific mood impact or a specific ailment alleviation. So it increases the complexity of doing these studies exponentially. So now that you have some background information, hold on, we're going to shift gears and talk about kind of how we get from there to, from a raw plant to individual compounds and how we can study them. So the first discipline really is separation science. How do we get pure isolated compounds that we can then test in cells? It's important for them to be pure because we want to know the impact we're seeing is from the known agent and not some unknown agent. So we need them pure. Botanical extraction has been around a very long time. It's well established for many commodities: oils, fats, waxes, starches, proteins, terpenes, even pharmaceuticals like insulin and vaccines are made using industrial scale type, very well developed technologies. So what's involved in separation science? To get pure compounds, we need to go through kind of a stepwise purification sequence that usually starts out with some sort of extraction, which can be solvent-based extraction, where we're using ethanol, butane, CO2, whatever, or it could be pressed out, like with olive oil, heat and presses, or just liquid-liquid extractions. But then we're left with a raw plant extract, at which point we can then apply distillation techniques to enrich the goodies, the cannabinoids, even further to a high purity and remove undesirable kind of impurities. The next, this can be done with crystallization as well, like with sugar, you just crystallize sugar right out of it. And we also might do precipitation to clean up things like waxes from the CO2 oil. We'll winterize it and we're precipitating out impurities, basically to clean up the matrix, the sample, so that we can apply it to chromatography. Chromatography is the most powerful technique, and it's been around a long time, since the early 1900s. And in chromatography, this column chromatography, you basically have a column filled with sand, and it's very special sand. It's silicon dioxide. The sand is all the same size, and it has special surface chemistry. So just for ease, the sand is immersed in a liquid called the mobile phase. The sand itself is called the stationary phase. It's in the column, and we flow a liquid through it, the mobile phase. We will dissolve a mixture of compounds, say it's a distillate, and it's CBD, THC, it's some, CBG, something like that. And we dissolve it in the solvent, it runs through the column. Each of these compounds interact with the stationary phase a little bit differently, and so it changes the rate. They all flow through the column at a different rate, and they separate into bands. As these bands flow through the column, we can collect them individually. We call this fractionization. So every 10 minutes or so, we apply a new plug of mixture. It goes through the column, and the beakers fill up with pure compounds. So we do this over and over throughout the day, and we get full beakers of purified compounds. So, a closer look at the media, it's just sand, very special sand, very expensive sand, and we can do stuff like derivatize the surface of the sand. So if we zoom up, we see they're perfectly round, very even and even particle size distribution. If we zoom up further and look at one, these are really like pin cushions. So we have a C18, 18 carbon molecule sticking out from the surface. So envision this as a pin cushion, and there's hundreds or thousands of these sticking out from the surface, and then there's hundreds of thousands of the particles themselves in the column. If we zoom up even further, we see the C18s are like little threads, and the cannabinoids or analytes flow through this in the mobile phase, and they interact with these threads differently. And in essence, that is the mechanism of separation. So if we apply modern technology and mechanics to this, there's several types of chromatography. Thin layer chromatography is just silica on a glass plate for doing very quick qualitative assessments of what you have there. We have HPLC, which is column chromatography at high pressure. We use this a lot to analyze what's in our fractions and in our extracts so that we know what we're dealing with. And then there's flash. This is like HPLC, but lower pressures, higher volumes, less expensive media so that you can load more sample for purification throughput. There's SFC, supercritical fluid chromatography. This uses supercritical CO2 as the mobile phase, which makes it very, very green, but the equipment is very expensive. Once it's up and running, it's a very green, very powerful technique. The last one we have in-house is called centrifugal partition chromatography. This one's really high-tech. You take two immiscible liquids and you pump one through the other, and it does chromatography. The systems are really quite simple. It's reservoirs of the mobile phase, a pumping unit, a unit that puts it's got a little loop where you can fill your sample in, and it introduces that sample plug into the flow path. It goes through the column. They separate, and then it goes through this detector, which is just it shines a light beam through a flow cell, and when compounds absorb the light, it makes a little peak on this chromatogram. Once we see the peak, we use a robot then to transfer the outflow to these various vials and containers, the fractionation. So at the end of the day, every 10 to 15 minutes, we're injecting a plug, and these bottles fill up. We get purified compounds. So there's a couple kinds of chromatography, normal phase and reverse phase. This basically refers to the stationary phase, whether it's polar or non-polar. So what does polar or non-polar mean? Well, in a molecule, a non-polar molecule won't orient in an electric field, meaning the charge distribution on the molecule is even. Whereas a polar molecule, the charge distribution is separated. There's some atom that likes electrons more, and it makes one side of the molecule a little bit negative, which makes the other side a little bit positive. And these will orient in an electric field. This polarity is kind of based on how it's bonded, whether it's an ionic substance like salt or a covalent substance like wax. This determines how evenly the electrons are shared in the molecule, and then there's some in between. This enables us, this gives us the separation handle. So the difference between a non-polar compound and a polar compound gives us the ability to make chromatography work and give us the selectivity to separate a whole host of pretty much any compound you can imagine. So, examples of chromatograms. This is what we actually see when we're processing a hemp plant material. We start with a raw hemp extract, and this was an cold ethanol extract, and we enrich the cannabinoids to about 65 to 75%. Now what's interesting, you see all these peaks. Each one of these peaks represents a compound. And remember, it went through a flow cell, the light was absorbed, and we see the peak. So this was undecarbed, but we can tell just by looking at this chromatogram, there's acid, CBDA, and neutral, CBD. This means this plant material sat somewhere hot and decarbed. If it were fresh off the field, we would see half as many peaks. They'd all be the acidic forms. So then we take this extract and we run it through a white film distillation system. This one is a VKL root system. And we enrich the cannabinoids about another 20%. And in the course of this, it's a heated process, so we decarbed all of them. So you can see the same extract went from over 20 peaks to like maybe 10. So we've converted most of the acids to neutrals here, and we can see we've done our work. What we do then is run it through CPC, the next stage in the purification, the chromatography. And this is one of the fractions that comes out. You can see it's very, very pure. It's 97% cannabinoids now. 99% of them are the CBD. So what's interesting about this fraction, this will spontaneously crystallize and drop out the CBD. So then we're left with 100% CBD, and all these little peaks remain in the solvent. So we dry that down, and now we've got an enriched feedstock with these very rare, low abundant cannabinoids like THCV, CBGV, all of the variants, and little unknowns, things that don't line up to any standard, things that we don't know what they are. These are very exciting because we collect them, test them in cells. If they have activity, then we send them out to find out what are they. So the importance of the unknowns is there could be something, a very minor component that could have activity that imbues a major impact on the therapeutic effect. So there are still quite a few unknown actives in cannabis extracts, and literally thousands of scientists' lifetime of work to find them all. So this is very nascent and early. We haven't learned very little compared to what there is to learn. So now let's talk about the importance of formulation. How do you get this stuff to work? So a pharmaceutical formulation is basically active ingredients and non-actives combined to make a medicinal product. There are various levels of technology involved in making a drug formulation. There are some very no-tech products. This is basically a solute dissolved in a solvent like sugar water. Anyone can do it. It doesn't take much technical prowess at all. They work well for certain compounds, but not well for hydrophobic, water-fearing, non-soluble, low bioavailability compounds. It really doesn't do much, meaning it's not bioavailable in this form. Then we have low-tech products. So low-tech might incorporate a tertiary solvent system like alcohol, PEG, or VG, and to make an emulsion or make a dispersion. There is a little bit of increase in bioavailability for hydrophobic compounds, but still not very much. It takes a little more knowledge to make these kinds of products. The next tier would be the high-tech products. This uses encapsulation, micro or nano encapsulation or micro emulsions, and it requires specific knowledge and equipment, and its aim is to increase the bioavailability of your therapeutic. The highest kind of level would be defined as controlled release. Now, controlled release can be fast action, delayed release, sustained release, modified release, composite release. If, say, your active degrades in stomach acid, you have to protect it till it can be absorbed in the small intestine. All of these things require intimate technical knowledge and equipment and very elaborate assays to be able to measure these dose forms to make adjustments, so to get the numbers right. Very expensive and very, very difficult to develop. So where are we at today in the industry? We're really at these two levels. There's a few people digging into the high tech. You see some microencapsulated products, but most are really low tech or no tech products that you see for CBD out there. To me, this is a slight problem because it tends to be a profit-driven endeavor instead of more altruistic motivations. So, how do we increase bioavailability of non-water-soluble things, or in other words, hydrophobic things? So a hydrophobic compound is one that is water-fearing. It's soluble and likes to be with oils, waxes, butane, heptane, organic things, non-polar things. Hydrophilic means it's water-loving and it dissolves in water. That could be like alcohol, it's miscible and dissolves, salts dissolve, vinegar, things like that. So they're readily miscible and dissolve in water. So how do we make something that won't dissolve in water get into a cell? We use technologies, using me cells is probably the most popular way right now. If we look at a plasma membrane of our cell, it's made up of a bilayer of what's called phospholipids. Phospholipids are kind of neat. They have a polar head and a non-polar tail. So the head is hydrophilic. It likes to be around water. The tails are water-fearing. They hate to be around water. So when you put them in a solution, they spontaneously form this thing called a me cell. The me cell has all of the hydrophobic, those are like the long C18 chains. They all kind of congeal together in the middle and have the hydrophilic heads face the water. This is what it would look like in a computer rendering of an actual me cell. So all this stuff, the green, it's hydrophobic. It doesn't like water, so it distances itself from the water, and it has a shell of hydrophilic heads that do like the water. I can dissolve CBD, THC, any non-water soluble thing. I can actually incorporate and get it to dissolve right in the middle of this me cell. This me cell could be a different size. It could be 25 nanometers or 25 microns. So the size of the me cell determines how much drug payload you can put in there. So all of this technology is fairly well established, and it's being embraced in the cannabinoid space for about 2 years now. We're going to start seeing a lot more high-tech products coming out the pipe. The reason we do this is if you look at the cellular membrane and you look at a me cell, they kind of have a similar structure, if you will. And so these me cells can go right through cellular membranes or right between them. They're very miscible. It gets it into the cell and into your system. So this is one of the best strategies. Anonymization, all these strategies work for increasing bioavailability. So once we get it in, once we figure out what we have, what it does in the cell, how do we get it in, and then what do we do with it? So at EBU, we wanted to predict the impacts of poly-pharmacology, in essence, map the entourage effect. And so traditional pharmacology deals mainly with monopharmacology. It uses a single receptor with a single drug agent to treat a single ailment. With cannabinoids or cannabis, it's really poly-pharmacology. We have multiple receptors involved, multiple active agents. So it becomes much, much more complicated. So much so that, let me go back. So why is this important? What we try to do is we start at separation science with pure compounds, and then we might test them in cells to get responses. We'll formulate more than one active compound in maybe a particular ratio. We'll test it in the cells. If we see something interesting, we'll test it in people. And we were doing this under the medical marijuana legalization. And we would test it in people in vivo, and they would respond as to how it made them feel. Then we might reformulate and go back and test it in people, get their psychopharmacological answers. The important thing is this link between cellular pharmacology and psychopharmacology, the pharmacology. No one has really ever tried to establish what we see in the cell activity to the mood effect in a person. The research has been illegal until just recently. So there's literally thousands of lifetimes worth of work to map the entourage effect. It's very, very difficult, and it's iterative and takes literally years to just get a few data points. So that's our goal in hopes of formulating products that have predictable, reproducible effects. So I'm not good at math, I took a shot at it. Billions and billions of combinations. So if we look at a monopharmacological approach, one receptor, one ratio of one compound, we can vary the ratio from 0 to 100%. We can also have 0 to 100% activation, which gives 10,000 possibilities. Now within that, there's only a small subgroup of domain that is actually pharmacologically interesting, and the rest are just kind of background noise. But if we increase the number of players to say 2 receptors, 3 ratios of 4 compounds, say 2 terpenes, 2 cannabinoids, now the numbers truly get astronomical. We're talking 10 to the 29th. So again, a lot of that is background noise, but we're talking about massive volumes of individual assays that need to be tested to see if there's anything there. So big black box, and we're getting little points of light every time we see something interesting in the cells that correlates to a great effect in people. We've also seen not so great effects in people, where a particular terpene will increase anxiety across the board. And this terpene is prevalent in all strains of marijuana, and hemp. In fact, it's probably the first or second in abundance. So we found if we didn't include this terpene in formulations, anxiety never increased. So with this terpene, anxiety always increased. So it's little data points like that that will slowly combine to give us an ability to map. And we have a lot of work to do. So now I want to talk about nomenclature a little. There's a lot of confusion around the nomenclature. Whole spectrum, full spectrum, broad spectrum, isolate. All these things really need some qualifiers to accurately describe each one. So a rose by any other name, it's still a rose. What do you call it? On the left here, I have all the different types of methods you might use to extract. And on the right, I have kind of what we use scientifically to describe things. So the strain name is completely arbitrary. It's basically assigned by the guy that grew the plant, developed it. And it might have things that, like the indica, sativa, hybrid, or Bubba Kush, whatever. But it's completely arbitrary. And it might relate to how the plant looks, the phenotype. Does it have fat little leaves or long skinny leaves? And people attribute that to indica or sativa, and that's how it looks, the physical kind of attributes of the plant, which are based on the genotype. And that is what genes are in the plant that could actually be expressed to produce that trait. Just because they're there doesn't mean they're expressed. It's environmental. How the plant was grown, it fully determines the chemotype, which is what I care about as a chemist. That means after I chop that plant down and look at the compounds, what do I see? So for me, the other three are interesting, but they don't really tell me what I'm working with. Only the chemotype does. So if someone says, hey, I've got this great full spectrum extract, well, a full spectrum what? Full spectrum dry sieve, a full spectrum CO2 extract, full spectrum ethanol extract. Each one of these types of extracts will produce a different chemotype. So without qualifying what your full spectrum is, it's kind of inaccurate. Full spectrum CO2 oil, great. So with that being said, what is a whole spectrum? A whole spectrum would be you shove the whole plant in a juicer and drink it. So it's got the cellulose, stems, roots, dirt, and all. Whole spectrum. A full spectrum would be, a full spectrum ethanol extract would be all the chemicals it extracted using ethanol. A broad spectrum might be, say you start with a hemp extract that's 0.3% THC, but now you've extracted it and it's 3% THC as the oil, and I want to get rid of it. So I run it through a process, mitigate the THC, and I've removed something. So I've taken the full spectrum and reduced it to a broad spectrum CO2 extract or whatever, because I've taken something out, but there's still an array of chemicals. And the next one would be a true isolate, where it's one chemical, it's in a crystalline or resinous liquid form, but it's pure. So that's important. When you hear someone say full spectrum, you need to say full spectrum what? Otherwise, they're kind of feeding you minutia. So, psychopharmacology. What is psychopharmacology? It's basically, does the drug or agent have a psychotropic effect? Meaning, does it affect the mind, the mood, the behavior, consciousness? We know ones that do, THC, alcohol, opiate, we can see the changes in behavior and the impacts on the mind. A lot of psychiatric drugs are psychotropic, like benzodiazepines or anything that gives you that great euphoric feeling you want. These are all psychoactive types of responses. They have classified different types of psychoactivity. Is it euphoric, dysphoric, stimulating, depressing, calming, anxiety provoking? What's funny is this Venn chart overlaps all the different types and the drugs that fall within these types. Cannabinoids is right here, right in the middle. So I guess it's all of them, My experience is I've never gotten depressed from cannabinoids. So this chart just is kind of a idea of the types of psychoactivity that are involved. So is CBD psychoactive? Well, people say it's not. I think it's because it's not bioavailable. If you take CBD in a bioavailable form, I think you can feel it. I can feel it. So I think it's somewhat psychoactive. So the cosmic debris, we'll talk about this. We've all heard about what CBD can cure and the claims. And a lot of it is lore, but where does the lore originate? It isn't just someone sprinkled pixie dust in the air and now everybody believes this. The lore originates because it had some impact on somebody and they're talking about it. They're providing anecdotal, experiential information about what it did for them. And there's a lot of different indications. What's neat is eventually the lore becomes real science, like with epilepsy. GW Pharma came out with Epidiolex, and so the lore that it helps epilepsy is not lore anymore. It's been proven scientifically. Same with Sativex. This is an antiemetic, anti-nausea drug. It was lore, hey, cannabis helps reduce nausea. Well, now it's science and it's fact because they did the studies and have a drug that showed that this lore is now fact. So other indications, pain. Pain's a huge one. Pain's a very difficult one because pain has redundant biological pathways. Pain is a very important feeling to express for survival. So if you cut off one biological pathway for pain, there's another one that gets rerouted, like Wesley on the Enterprise man. He just reroutes that to another biological pathway, and you still feel the pain. So it's much, much harder indication, and it will probably take multiple agents to really get pain under control without using opioids. The other big one is opiate use disorder. I've read some studies lately where people that do heavy cannabinoid regimens during their withdrawal time course have an 80% less recidivism rate to go back to using opioids than people who didn't use cannabinoids during their withdrawal time course. So I can see it being used big time in opiate withdrawal treatment in the near future. And then there's a whole host of others, depression, Parkinson's, Alzheimer's. I've heard pretty much anything you could have that CBD will cure. One interesting note, obesity was also an indication. They came out with a drug called Chromatography, and it was a CB1 inverse antagonist. It was pulled from the market because although it worked at keeping people from getting obese, it caused suicidal tendencies that resulted in several deaths. So it was pulled from the market. Again, now it's not lore. So what am I buying and consuming? This is my favorite slide. These are absolutely not in any way affiliated with me. I'm not endorsing them. They are simply examples I pulled by typing CBD into Google. And this is what I came up with. I pulled some interesting products. We have Moon Juice Sex Dust for $38. I have no idea what it does, but it's got a neat name. And then we could buy ourselves a carton of hemp cigarettes for $80. Again, there's nothing there that tells me what percentage of CBD was in the hemp. So this is just kind of a novelty. This one, these guys got a little jiggy. They're talking about the endocannabinoid system, so they're throwing a little technical jargon your way. And it's only $40. This one I love. There's 30,000 milligrams in 120 gummies. Man, that's a lot. That's 250 milligrams per gummy bear. Wow, that's a big dose of anything. So I love it because they're really highlighting how much is there. And then the hemped up soda. This one I like. It's only $50 for a 12-pack. So. This one probably has the most potential to have increased bioavailability because to get it into a beverage, they had to do something to make it soluble. So of all these products, this one's probably got the most tech because it's in a liquid. These others are no tech products, 100%. And if any of these are your products, I'm sorry, they're just examples. So how do you determine the best value? I put a little example here. We got $38 bottles of CBD. This one advertises 50,000 milligrams, but it has no technology. So its bioavailability is 6%. That means only 3,000 milligrams actually gets into you and gets used. Here we've got one that's only 10,000 milligrams, but it's 50% bioavailable. So that means 5,000 milligrams is actually utilized. Which one's the better value? The one with less, not the one with more. So these are the kinds of things that people just don't have any education or realization that the formulation is really everything as far as the value of the product. So, why do humans self-medicate? My favorite self-medicator was a guy named Sasha Solzhin, who developed MDMA in the 70s. And he used it for very powerful therapeutic. He used it in coordination with psychotherapy and saw profound results. And then it was made illegal. He wrote, he was a self-experimenter, so these drugs he made, he used, and he wrote a book called PCAL. It's a great read, and it stands for phenylethylamines I have known and loved. Okay, I'm done, I'm being told. Then Bobby, one more. Bobby self-medicates because, you know, he reads the little labels on the bottles and he knows how he'll feel the next moment. So that's another reason. Okay, I have to do one more slide. Takeaways. Beware of the mania. Get educated. Experiment. Find your blend. So terpenes, CBD, THC, everyone has a different blend that does something for them. Find yours, stick with it. Know why it benefits you, and is it contributing to your well-being? Lastly, Project CBD. It's a great website. It's reputable information. If you take other medications, you want to read this cannabinoid drug interaction pamphlet. It's very important. And I want to acknowledge my colleagues that helped us get to where we are, especially Chris Denicola. Is he out there? No? Okay. Anyway, thank you very much for your time.